Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Mary Phillips will present Developing New Treatments for Mania Using Brain-Based Risk Markers. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $430 million to fund more than 6,200 grants around the world. I'm delighted to introduce Dr. Mary Phillips. Dr. Phillips is professor in psychiatry and clinical and translational science at the University of Pittsburgh School of Medicine. She was a 2005 BBRF independent investigator, the 2017 Colvin Prize winner for outstanding achievement in mood disorders research, and she's a member of our scientific council. Today's webinar will begin with Dr. Phillips' presentation. This will be followed by a question and answer period. To submit your questions, please use the questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as possible in the time allotted. And now, I'm pleased to introduce Dr. Phillips. Mary, the floor is yours.
Thank you very much, Jeffrey, and thank you everyone for being here today. I just want to say it's a huge pleasure for me to be able to spend a few minutes presenting some of the work I and my colleagues in my labs are doing, and I'm very excited to share this work with you today. So I first of all just want to start off by talking about some of the take-home truths about bipolar disorder, this disorder which is so debilitating and so devastating for so many sufferers around the world. It is a very common illness. Most people don't realize how common this illness is. As you can hopefully see on my first slide here, we see the figures shockingly high of nearly 5% lifetime prevalence in adults, even very common in adolescents. As many of us know, and I know many of you in the audience will be all too well aware that sadly this disorder is characterized by huge difficulties in regulating mood, where people unfortunately have to suffer the consequences of having very high, elevated, dysregulated mood, i.e. mania, but also the terrible lows of depression. And it's these dreadful mood swings and the inability to regulate and control mood that cause the problems with the switching, so that people with bipolar disorder are often suffering from the highs, lows, often both together without any real ability or capability of controlling their mood appropriately. Now, mania, of course, is the characteristic symptom of bipolar disorder. Unlike other mood disorders such as major depressive disorder, bipolar disorder has the mania as well as the depression. So if you will, it's almost like a double hit. Not only do people suffer from the terrible lows, but the high moods are also debilitating. I just want to highlight how debilitating the mania is. This is not a fun thing to experience, despite what many of us hear in the popular press and newspapers. Being mania, having a problem with mania, being manic, even having the little mania, hypomania, is not a healthy thing, and it can cause all sorts of problems, as I'll allude to later. It shockingly is the fourth leading cause of non-communicable disability and disorders worldwide. It's expected to take approximately 10 years off the lifespan, and there's a 20 to 30 times greater suicide risk than the general population. So this is an incredibly debilitating disorder. Now, why haven't we got any further with helping people with bipolar disorder? Well, a huge problem with bipolar disorder is that it's very challenging to study because it's so complicated. And a problem is that we have no objective brain-based biological markers that tell us ahead of time which people are going to be most at risk for developing the disorder. And this, of course, makes it difficult not only in identifying people who are going to be at risk for the developing the disorder later, but it also doesn't allow us to make any strides forward in developing new treatments, because unless we know what the brain basis of this disorder is, it's very difficult to develop new brain-based treatments. And this is what I want to talk to you about today. But first of all, I just want to highlight where we are at currently with treatments for bipolar disorder. There are, of course, many existing treatments, sometimes quite effective, but mostly the treatment tends to rely upon using medications, psychopharmacology. And unfortunately, because bipolar disorder is so complicated, involving not only mania, but also depression and the mood swings between the two, multiple medications often have to be used. So I know of many of my patients and colleagues even who have bipolar disorder who often are taking 4, sometimes 5 different medications and then some. Very, very difficult to treat. Unfortunately, with more medications, the more likelihood there is of having unpleasant side effects. Not all medications are tolerated, so even if one type of medication works for one person, it's not going to necessarily work for somebody else. And not all medications work for everyone. Lithium being probably one of the most effective treatments for some people for mania is not effective for everyone. And for some people, the side effects are so awful that it's just not worth taking. So because of all of this, it's often and frequently really difficult to prevent relapse of mania or depression or both in people with bipolar disorder, simply because the medications don't work. Even other types of treatment, psychotherapy, behavior therapy, often work only in certain contexts and they need the medications there to help them work. So most people with bipolar disorder unfortunately have to rely upon a cornucopia, as it were, of different types of medication. And as I've just indicated, oftentimes these medications are not optimal at all. So if only we could find the spot in the brain, the circuits in the brain, the parts of the brain that lead to this mood instability, emotion dysregulation. But if we could do that, we could start to not only prevent mania, but in so doing, prevent the switching from mania and depression down the line. And as we all know, people with bipolar disorder who have problems with regulating their mood, medications for whom medications don't work very well, over time, the inability to regulate mood and the terrible switching between the moods gets worse and worse and worse. So it's critical to try and understand the mysteries of the brain and how this goes wrong in causing mood dysregulation in people with bipolar disorder as soon as possible, so that we can detect earlier people who are at risk with the disorder and also start to develop new treatments with the knowledge of what's going wrong in the brain. So where do we start? Well, I want to highlight one aspect of bipolar disorder, which many people understand, but perhaps not everyone thinks about all the time as a critical part or problem of having bipolar disorder. And that is this high reward sensitivity and high ability to expect reward, and how this relates to impulsivity and sensation seeking. So let me talk you through this slide. So people who are sensitive to reward and who are expecting to get rewards in life often are quite impulsive. So, as soon as there's a context when they think that there may be a possibility of being rewarded, whether it's with money or a job promotion or a love interest, a new house, etc., as soon as they're in that kind of situation when there's a possibility of something good happening, they become impulsive. They start to be at risk of performing impulsive acts of all descriptions, taking risks with medications, taking risks with substances, money, etc. Related to the impulsivity is the sensation seeking, which is this willingness to actually take risk and seek novel and intense sensations and experiences. So the combination of these two components, this impulsive way of being and this wanting to seek and take risks for novel sensations and experiences together can be highly debilitating. And we know that this combination of features actually is evident in people who suffer from bipolar disorder. Moreover, we know that in young people who have not yet developed a formal diagnosis of bipolar disorder, they also are predisposed to having this impulsivity and sensation seeking. So it's there in people who haven't yet got bipolar disorder, but who are at high risk of getting bipolar disorder. And this impulsivity and sensation seeking is also there in people who already have started to develop the first signs of bipolar disorder and get the diagnosis. So this, I believe, is a key set of behavior traits or response tendencies, if you will, that can help us, give us a clue as to where things might be going wrong in the brain of people who are at risk for and those who already have bipolar disorder. So what to do in the talk today is, first of all, talk about how we in my lab are using different types of neuroimaging to try and understand the brain basis of the development of this impulsivity, the sensation seeking, and therefore risk of becoming, having a diagnosis of bipolar disorder later on. And then I'm going to talk about how we're using this knowledge to actually identify new brain-based targets to develop new, we believe, safer treatments for bipolar disorder that don't necessarily have all the awful and dreadful side effects that the combination of different types of medications unfortunately does have. What I'm going to be talking about a lot today are regions in the brain reward network or brain reward circuitry. And here's just a cartoon showing just a few of the regions that are important. Now, this is not exhaustive, and I've simplified this just for the purpose of time and expediency today. Obviously, the rewards, the way our brains perceive and encode and anticipate reward as humans, it's very complex. But these are key regions, and in red in particular, I want to highlight in VS, which is the ventral striatum, the seat of reward and pleasure in the human and mammalian brain, and indeed non-mammalian animals as well. And then in blue, there are prefrontal cortical regions, or orbital frontal cortex, OFC, ventrolateral prefrontal cortex, VLPFC. I'm going to be talking a lot about that region. And these regions in particular work in tandem with the ventral striatum and help us experience the sensation of reward, experience the waiting for reward, generally regulate reward experiences. And in purple here are the arrows which really highlight the key neurotransmitter or neuromodulator, dopamine, which, as many people know, is a core neurotransmitter, linking these regions and transmitting information about reward in the brain. So let me start off by talking about how we're approaching this problem and how we're using multimodal neuroimaging to examine reward circuitry, conferring risks for bipolar disorder. What we do is we use a specific task which was developed by my colleagues Erica Forbes and Henry Chase. Henry Chase is in my lab, and Erica Forbes is another BBRF award winner in the past, and she works with me very closely in Pittsburgh. And we together have developed this task which we feel really tries to capture the problems that people with bipolar disorder might experience when they're in a context when they might have a possibility of gaining a reward later on. And I just want to highlight that this reward task really, I'm going to show you how it's structured, but this reward task allows us to examine how the brain works when we are in a context of expecting a reward, and then when the reward comes or it doesn't. And I just want to highlight the fact is highlighted there in the purple font that it's this reward expectancy, this anticipation of something good that might happen. This context we know triggers frustration in people who are more impulsive and reward sensitive, i.e. those who are more at risk of bipolar disorder. So focusing on this reward expectation or expectancy period is a good way to start to try and understand how the brain processes reward expectancy and how that in turn might predispose to bipolar disorder or to mania in particular. So this task has 4 types of expectancy. And I'm just going to talk you through this. As you can see on the screen, there are 4 arrows and cards on the left-hand side. Now this first context with the arrow pointing upwards and the cards shuffling. In this task, what this means is that if when you do this task and you get the answer right, it's basically a simple card guessing task where you have to guess whether a card to be shown is going to be higher than 5 or lower than 5. This tells you, this first context tells you that if you get the answer right, you're going to win money. If you get the answer wrong, don't worry, you're not going to lose anything. Now, the second context, you're anticipating again, this time the arrow's down. So you're guessing what the card might be. If you get the answer wrong, this time you're going to lose money. But if you get the answer right, nothing will happen. The third context is kind of a mixture, whereby if you get the answer right, you could win money. If you get the answer wrong, you're going to lose money. And the fourth context is neutral, nothing's going to happen. So this context of this task comprising 4 different anticipation contexts actually allows us to kind of measure how the brain activity in the reward circuitry, the reward network, varies as the expectancy of reward changes from a 50% chance of win, 50% chance of loss, 50% chance either way or nothing. So it allows us to gauge how the brain reward expectancy circuits might ramp up or ramp down as the potential to gain reward is there or not. So what I'm going to tell you first is about some of the earlier studies when we used a version of this task in people with established bipolar disorder. Now, when we started off this work, we had assumed that what we would see in the brain would be basically patterns of activity focused on that area of the brain I talked about earlier, the ventral striatum, the part of the brain that is known to encode reward in many different contexts. And we had expected to see that area of the brain in particular be the key part of the brain that might be higher in activity in people with bipolar disorder. But actually, what we found was something in addition to that. So whilst we found that the ventral striatum was indeed important in helping us expect reward in that high expectancy reward context that I just showed you, what also we showed is that in people with bipolar disorder, whether they had a diagnosis of bipolar 1 or bipolar 2, i.e. mania or hypomania, as well as depression, we found that an area of the brain circled in red there, and that's the left ventral side of the prefrontal cortex, ventrolateral side, so that's this left ventrolateral prefrontal cortex, that was elevated in activity in people with bipolar disorder relative to healthy people without a bipolar disorder diagnosis. So that was interesting. But at the same time, in another study, and at that time I was working both in Pittsburgh and also in Cardiff in the UK. So the first study was in Cardiff in the UK, and the second study, now I'm about to show you, was in Pittsburgh. We also used a slightly different structure in the sense that we, rather than just have people with bipolar disorder and healthy, we had a group of people who had bipolar disorder depression, a group of people in blue who had unipolar major depressive disorder depression, and then a group of healthy controls. So it's a slightly different group of people that we asked to take part in the experiment, but we used the same task. And what we showed was yet again, the people with bipolar disorder showed higher activity in the same left ventrolateral prefrontal cortical region when they were expecting the higher reward stakes. And this was in comparison to people with unipolar depression and healthy controls. So already 2 lines of evidence suggesting that there's something abnormal in part of the reward circuitry in people with bipolar disorder when they are expecting a likelihood of reward. And that was seated in this left ventrolateral prefrontal cortex. So then we started asking questions about, well, what happens if you don't yet have a diagnosis of bipolar disorder, but you are at high risk of developing bipolar disorder because you're high on that impulsivity and the sensation seeking that I was talking about earlier. So you're not yet with a diagnosis of bipolar disorder, but you are at future risk of having a bipolar disorder diagnosis. Well, here's another study done a few years later, and this is in a large group of people, over 100 people, young adults, 18 to 25 years of age. And they performed the same task that I just showed you. And what we found when we looked again at their whole brain is that, again, as they were in the context when they had a chance of winning reward, those people with higher levels of impulsivity and sensation seeking had higher activity in the left ventrolateral prefrontal cortex, the same region as I've just shown you in the people with bipolar disorder. In a next study, we then tried to replicate these findings in an independent group of young people, again, young adults between around about 18 to 25 years of age. So this is another sample of, and this time 127 young people. And we showed the same thing, that basically the more impulsive the young people were, the more likelihood there was to have this left ventrolateral prefrontal cortical activity this pattern of activity being there in the context of the likelihood of getting a reward later on, so the high reward expectancy context. And what we also found is that one part of impulsivity in particular seemed to relate to this pattern of higher left ventrolateral prefrontal cortical activity, and it was negative urgency, which is a facet, a component of impulsivity, which essentially means that when you're in a position of frustration or something you want to happen that isn't necessarily going to happen, such as expecting a reward and then it perhaps not happening, you tend to act rashly and impulsively. So this part of impulsivity in particular seems to be associated with this left ventrolateral prefrontal cortical activity. And what is more, we found that this relationship between left ventrolateral prefrontal cortical activity during expectancy of reward and this component, this key component of impulsivity, negative urgency, were in turn related to future risk of having a bipolar disorder diagnosis, as measured by this very interesting scale called the mood scale, which is actually developed by my colleagues Anne Frank and David Kupfer in Pittsburgh with their colleagues in Italy a couple of decades ago now. But just to highlight what we showed is we showed essentially that the more left ventrolateral prefrontal cortical activity during reward expectancy, the likelihood was that there was more negative urgency, which in turn related to more of this mood scale, a measure relating to bipolar disorder risk. So there we are showing this sort of three-part relationship. And very recently, and these are actually data in preparation for submission very soon, what we've shown is that even when you measure the bipolar risk, bipolar disorder risk over the course of a year, so rather than having one measurement in time, you measure it when you do the scan, you measure it 6 months later, and you measure this risk a year after that, so over a whole year. And when you take the maximal measure of bipolar disorder risk, we still see this relationship between left ventrolateral prefrontal cortical activity to the reward expectancy and this measure of future bipolar disorder risk. Now, what I've shown you so far, of course, is using fMRI, functional magnetic resonance imaging, to measure neural activity at the sort of larger scale macro level. And as many of you know, functional neuroimaging, fMRI, is an excellent tool, and it allows us to kind of look at neural activity in the human brain. But what it doesn't do is allow us to have high temporal resolution, so high temporal levels of sensitivity. What EEG can do, which fMRI cannot do, is allow us to kind of have a proxy measure of what the underlying neuronal level activity is, sort of neurophysiologic level, at a very high level of temporal resolution. And here's a picture of my colleague Dean Salisbury, who kindly showed us what it looks like for him, at least, to put on this this headgear, which allows us to measure his EEG brainwave signals, if you were, when he's doing a variety of tasks, including this reward expectancy task. Now, what we know about EEG and the ventrolateral prefrontal cortex region that I'm interested in, and my lab's interested in, is that the VRPFC is, for our purpose, fortunately, very sensitive to be measured by EEG because it's got relatively thin intervening tissue between the skull and the brain over this part of the human cortex. So it's actually an appropriate region in which to measure EEG signals. Now, we also know from a host of other studies outside of our lab that prefrontal cortical activity in a particular frequency, EEG frequency, the beta and gamma band, which is relatively high frequency, is associated with reward processing and reward expectancy. And that this power, this activity in turn reflects underlying dopaminergic activity. And if you remember in one of the earlier slides I showed you, those purple arrows, the dopamine clearly being a key neurotransmitter in reward circuitry. So taken together, it makes sense as as a parallel step in combination with the fMRI to look at EEG, particularly EEG measures of ventrolateral prefrontal cortical and other cortical activity in the beta band, because that tells us not only about neuronal activity, but gives us a window into understanding what underlying dopaminergic activity in these regions might be doing. And we also know from a couple, not a large number, but a couple of existing studies, is that all of the above, the beta activity in the prefrontal cortex, appears to be associated with facets of this impulsive sensation seeking that we know is in turn associated with bipolar disorder. So what we wanted to do is ask the question, can we put this all together and determine whether left ventrolateral prefrontal cortical activity during reward expectancy and activity specifically in the beta band is associated with the risk of bipolar disorder by way of measuring impulsivity facets. And essentially, this is what we've been able to show. So let me just talk you through this slide. This is led by Brian Kaufman, who works with Dean and myself in the Department of Psychiatry. And what Brian was able to show in a group of healthy young adults was that during this task, and here I'm showing you the four contexts, so this is the possible win, where you can either win the reward or not. You either have a mixture of win or loss, nothing happens, or you can lose or nothing happens. We showed that only in the context where there was this possible win, when you could actually potentially 50% chance of winning something down the line, so that high reward expectancy context again, only then was there this pattern of left ventrolateral prefrontal cortical activity in the beta frequency band is shown here. So this is showing you the sort of beta activity, this is the frequency, and this is sort of the beta range here. And we've shown that this was specific to the possible win context, but also that it related to sensation seeking, thrill seeking, and as shown by these graphs here. So the more beta power over the left ventrolateral prefrontal cortex in this high reward expectancy stakes context, the more of that, the more likely you were to be impulsive sensation seeker. And of course, that relates to bipolar disorder risk. So we now have two measures, different types of brain measure of activity in the left ventrolateral prefrontal cortex during reward expectancy that relates to impulsivity and hence bipolar disorder risk. But the big question is, what is the left ventrolateral prefrontal cortex doing? What does it do? What does it mean to have high levels of activity? We know obviously it's associated with impulsivity and bipolar disorder risk, but what is it really doing? So I just want to highlight two studies which have been performed outside the context of psychiatry, but really just to illustrate what this particular region of the brain is doing in reward contexts. So the first context in which left ventrolateral prefrontal cortical activity might help us understand, its role in reward processing is reward expectancy and trying to obtain a large reward as possible in the future. So let me just talk you through this. Imagine you're in a situation when you are told you have a chance of getting a nice monetary reward in the next 10 minutes or so if you guess right, if you choose the correct symbol. And you have to choose between a circle and a square shown in red here. So you're thinking, gosh, which one am I going to choose? Is the circle going to give me the big reward? Is the square going to give me the reward? And then you find out that actually it's not that simple, because what happens is that the circle doesn't predict a reward. What it does predict is a high chance of it predicting a green triangle. But then that green triangle gives you a very high chance of getting a good reward. Whereas the square will give you a much higher chance of getting the smaller purple triangle, which will give you a lower reward. Now, you don't know any of this when you start off, so you're just choosing randomly. And then you find out by trial and error that the red circle, okay, that gives me the green triangle. And that green triangle, you then find out gives you the reward, and the same goes here. So ultimately, you're going to pick the circle because that's going to give you the green triangle, which in turn gives you the high reward. But how do you learn that? It turns out that the left ventrolateral prefrontal cortex, and this is the left, by the way, the left switched on this slice, but this is actually representing the left side of the brain. It turns out in another study performed by my colleague and friend Tim Behrens' group in Oxford, University, UK, his group showed that this region actually makes that critical link between the signal that ultimately is going to give you the high reward and the intermediate signal, the green triangle. The circle itself does not tell you you're going to get a high reward. The circle itself tells you you're going to have a green triangle, which in turn gives you a chance of reward. And the left ventrolateral prefrontal cortex appears to make that initial connection, which is very important because in day-to-day life, we often have to go through several stages before we get a reward. We don't just sort of choose one thing and then we get a reward. We often have to choose A, which leads to B, which leads to C, and so on. And down the line, we get the reward. So it's critical that we have our brains working so that we make these decisions appropriately. And that's what the left ventrolateral prefrontal cortex does. So take-home message here is left ventrolateral prefrontal cortex allows us to make decisions to optimize the chance of having a maximal reward. So that sounds good, doesn't it? But there's another point about the left ventrolateral prefrontal cortex which might not be so good. And here's another context. Imagine you're in a situation when you have to choose between a small amount of money now, $10 now, or a larger amount of money, $100 in a month. Which would you choose? Well, I suppose it depends, first of all, how much money you need and how much of a risk taker you are and or indeed how patient you are. Because if you're very patient, you're likely going to wait a month to get the $100. But it's known that people with bipolar disorder tend to make more impulsive choices. So they're going to be more likely to choose the $10 now rather than the $100 in a month. And what is also known is that the left ventrolateral prefrontal cortex, a region very close by the region I was showing you on the previous slide, promotes this choice of immediate smaller rewards over later larger rewards when you have to make a decision. So what we see so far is that the left ventrolateral prefrontal cortex in reward expectancy decision-making context allows us to kind of ramp things up to try and make links to optimize rewards, but it also, if we're not careful, tends to predispose us to going for, well, let me get the reward as quickly as possible. Okay, I know what I need to do, but let me get the reward, any reward now as soon as possible, rather than being patient and waiting for it. What I also want to show you is how we measure impulsivity in the lab behaviorally, and so that we can have this behavior measure of impulsive, going for the sooner reward and the smaller reward sooner rather than the larger reward later, and how can we relate that to the brain activity. So what we can do here is on the x-axis in this graph, I hope you can see here, we've got a delay. So that larger reward, you wait, you don't have to wait at all, you wait a month, 2, 3, 4, 5, 6 months, and so it goes on. On the y-axis here, I'm showing you effectively a scale which tells you the relative value of the later reward versus the immediate reward. And the lower the number, it means that the less valuable to you is the later reward. So the less likely you're going to wait for the later reward. This is called discounting. It means that if you're going to discount the later reward, it means that it's less valuable for you to wait for the later reward than it is to go now for the immediate reward, even though the immediate reward is smaller. And what we see is that in people who are very impulsive, it only takes a very small delay, like 1, maybe 2 months or even less, before they give up. They're not going to bother to wait for the later reward, no matter how large it is, and they're going to go with the sooner reward now because it's smaller. It's smaller now, and they're going to go for that. Whereas people who are a bit more patient, who are less impulsive, might say, well, wait a minute, I can get $100 in a month. I'm going to wait, and I'm not going to devalue, discount, that larger reward. I'm going to wait. And so they're less likely to ignore the later rewards and go with the immediate reward. And the key take-home message here is that we have this parameter called K, which is essentially a behavior measure of how impulsive people are in making choices about the smaller versus the larger reward, the sooner, smaller, larger, later reward. The larger the K, the more impulsive people are. So the larger the K, the more likely you are to have this red behavior curve, the more likely you are to discount the later, larger rewards, the more likely you are to go now with the sooner rewards. And that, of course, characterizes people with bipolar disorder. So what we did with Henry, Chase, and others in my lab is we designed a task. It's called delay discounting, because that simply means, you're trying to gauge the point when people discount, they stop wanting to, being patient enough to wait for the later, larger reward, and they go with a sooner reward. So this task is relatively straightforward. I'm just going to highlight two parts of it. Essentially, the first part is there by that blue arrow. There is a choice between $20 now, let's say, or $30 in 14 days. Now, that's not probably a difficult choice. I imagine that most people will probably go for $20 now, but imagine it could be different. It could be $2 now and $100 in 14 days. And people might actually decide to go wait for the blue and go for the $100. So we can mix it up, and there's a whole range of different values we have. There's also a whole range of different delays as well. So you can range from a relatively small amount and a small delay to a large amount and a large delay, and vice versa. The key thing about this task is that we ask people to make a decision early on in the trial. So here they're making a decision, and then we ask them to wait. And then a few seconds later on, they're going to have this slide when they choose. Now, if you notice, there's no numbers here. There's just a pink square and a blue square. So people remember that the pink is the now, and the blue is the later, and then they make their choice. So this allows us to measure what's going on in the brain when they're actually deciding which one they're going to go for before they actually make the choice. And what did we show? Well, I'm now going to show you what we showed in people with bipolar disorder versus healthy individuals when they were performing this task. This is again data in preparation. I'm very pleased to say we've just got funded on a new foundation award to examine this task in the scanner and a task like this and with EEG. And what I'm showing you now is pilot data in a relatively small number, but very promising data showing you how valuable this paradigm could be when trying to understand how the brain is abnormal in its function in people with bipolar disorder. So here, what I'm showing you is two brain slices. On the right is people with bipolar disorder, and on the left is people who don't have bipolar disorder. And what I'm showing you in the green circle is the activity in the beta range that is elevated in people with bipolar disorder when they're in that critical decision-making point in the task. So when they're making a choice between the immediate smaller versus larger delayed, and when we know that they've gone on to choose the immediate smaller reward. So that, if you will, this activity is greater in people with bipolar disorder before they go on to choose the immediate smaller reward. So there's a clue there. But what we also need to look at is their K. Are they actually more impulsive anyway? And the answer is yes. As I said in the previous slide, the K value gives us an idea of how impulsive people are. And as you can see here, the people with bipolar disorder had a much larger K, i.e. they were much more likely to go with the smaller immediate rewards and choose those over the delayed larger. So in summary, on this beginnings of this work, we're showing that already that adults with bipolar disorder have greater left ventrolateral prefrontal cortical activity when they are deciding between the two choices, and that this seems to predispose them to choosing the immediate smaller rewards, i.e. being more impulsive. So what I also want to highlight before I go to the last part of my talk is just another type of neuroimaging that we're using to also look at the reward circuitry in the brains of people with bipolar disorder versus those who do not. And this is looking at white matter tracks in the reward and other circuitry. So here I'm showing you the reward circuitry again, and I'm also highlighting 3 white matter tracks that link a lot of the reward circuitry regions. So the unsynaptic fasciculus is one which connects regions in the prefrontal cortex and other regions that I'm not showing here that are important for emotion regulation. The forceps minor is in red, is quite an important large tract in the front of the brain, which connects all of these different blue prefrontal cortical regions together. Very important for regulation of behavior and regulation of reward. And perhaps most importantly of all, for our purposes for reward, is the cingulum bundle in green here, which connects all of these regions together in many ways, particularly all the prefrontal regions and other parts of the cortex in humans, which are important for emotion regulation. So these are critical white matter highways, if you will, or white matter tracks that connect regions and reward circuitry. Now, this is work carried out with my colleagues Emilia Versace and João Lima Santos, and this is work also actually under review at the moment in a journal. Essentially, what we wanted to do is, first of all, identify patterns of white matter that predict worsening hypomania in people who don't yet have a diagnosis of bipolar disorder, and then determine whether these same measures were evident, whether these same patterns of white matter were evident in people with bipolar disorder relative to healthy controls. I won't go into detail about all the white matter measures we can use because we just simply don't have time, but I just want to highlight one measure, which is fractional anisotropy, the FA for short, which is probably the most popular measure used in diffusion imaging, white matter neuroimaging, and it's a measure, if you will, of the microstructural integrity of white matter. In essence, it's a measure of how closely and nicely aligned all the fibers are together in white matter tracks. And the idea is that the higher the FA, the better. The higher the FA, that means that the more closely aligned the tracks are, the more likely your white matter tracks are to be working properly. And to cut a long story short, what we have found, first of all, in the people, the young adults who don't yet have bipolar disorder, but who are at future risk of getting bipolar disorder, and I can show you why in a moment, these adults showed lower FA, this critical matter of white matter structure, in 3 of those bundles. The left cingulum bundle, which I showed you before, connecting the front and back of the prefrontal cortical regions with other cortical regions, the right cingulum bundle, and also the unsynaptic fasciculus, connecting prefrontal cortex with other temporal cortical regions important for emotion regulation. Now, what we critically showed here is in these young adults was that those adults who had greater increases in mania 6 months later were more likely had lower FA in these white matter bundles. So the less integrity of the white matter tract in these white matter tracks, the higher the mania score 6 months later. And these were, as I say, young adults who had not yet developed bipolar disorder, but obviously are going to be more at risk of developing bipolar disorder because they're developing greater mania even in the course of 6 months. And these are just some statistics showing how highly significant, these are very highly significant p-values these relationships were. And this is in a group of 81 young adults. Now, in parallel, we then went on and asked the question, well, I've shown you that left and right cingulum bundle and right unsynaptic fasciculus abnormalities in those white matter tracks actually predict future greater worsening of mania in young adults not yet with bipolar disorder. What about adults with bipolar disorder? Well, here we show in a completely independent group of adults with bipolar disorder and healthy controls, using the same processing, the same kind of approach to our data analysis, is that they too, the people in bipolar disorder shown in orange here, also show lower FA in these critical 3 white matter tracks. And most importantly, they show abnormally low white matter FA comparison with healthy controls, particularly in regions in these tracks that we know are associated with emotion regulation. So we've got clues even from the white matter findings that people who are at risk of developing mania also show abnormalities in the brain in white matter tracks that connect reward circuitry regions. So finally, I'm going to go on to talk about what we think is really exciting new work when we're using our knowledge of neural markers that predict risk of bipolar disorder and associated with risk of bipolar disorder, how we can use these to develop new treatments for bipolar disorder and mania in particular. Now, I'm going to talk to you very briefly about some of these studies. And the studies we're really interested in are using different types of brain stimulation or neuromodulation techniques to try and tap into this neural circuitry and, if you will, down-regulate the abnormally high activity that we see in the left ventrolateral prefrontal cortex in particular in people at risk for bipolar disorder or risk for future mania. So the first type of neuromodulation technique that we used was transcranial direct current stimulation, or TDCS for short. Now this is a type of brain stimulation which is very easy to administer. I mean, essentially you can use something really quite simple as shown on the left, or much more complicated here. Essentially, this type of stimulation uses electric current, and it simply has electrodes where you're passing a very weak indeed current through the brain, trying to kind of allow the brain electrical activity to reset itself. You either down-regulate it or up-regulate it. And in a first study, we used this type of stimulation, this type of stimulation led to the left ventrolateral prefrontal cortex, the transcranial direct current stimulation. We're targeting the left ventrolateral prefrontal cortical region actually in the brain scanner when people were performing the reward task, which is the advantage with this type of stimulation. And I'm just showing you one slide here. And the take-home message of this slide, and this was work really led with me by Michelle Bertocci in my lab, and as well as Henry Chase, whom I mentioned earlier. We showed that when we were stimulating the brains of people with bipolar disorder, and also of people without bipolar disorder, when they're doing this task in the scanner, we showed that the more we could reduce the left ventrolateral prefrontal cortical activity during reward expectancy, the lower the irritable mood after they came out of the scanner. Now, as we all know, and many of us know, irritable mood is a hallmark feature of mania or predisposition to mania, as well as also being present in depression. So if we can reduce this measure of risk for mania and depression and risk for impulsivity, irritability, with our neuromodulation, that is indeed a promising start to developing treatments which might be able to help us do a better job at treating bipolar disorder. Now, the problem with transcranial direct current stimulation is that it's weak and it's a bit diffuse in the sense that we aim to target the left ventrolateral prefrontal cortex, but we cannot be sure that we are totally targeting that region. We're often, with transcranial direct current stimulation, targeting many other regions as well. So what we've now moved on to use is another type of brain stimulation, neuromodulation, called theta burst stimulation, or TBS for short, which is using magnetic fields and is much more focal, it's much more specific in its targeting of the brain regions, which is ideal for our purposes. And this is an example of the machine that we use, the TBS, or transcranial magnetic stimulation, it's its other named machine. And as I'm showing you here, what we can do is we can target the left ventrolateral prefrontal cortex quite specifically if we put the coil, the TBS coil, over the left VLPFC, as opposed to over a control region here, which is the left somatosensory cortex, SSS for short. And we can have two very different targets in a way that we cannot do so well with the TDCS. And I'm showing you again preliminary data, which is also part of pilot data for which we've now been funded by the NIMH to do a much bigger trial in people with bipolar disorder and healthy controls using this technique to try and affect mood and risk for bipolar disorder. What I'm showing you here is the basic findings about which we're very excited when we're showing that if we target the left ventrolateral prefrontal cortex versus this control region, we can actually damp down the activity and downstream connections between the left ventrolateral prefrontal cortex and the deep reward circuitry regions, particularly the ventral striatum. We can damp down that connectivity much more in people with bipolar disorder when they're in the remission, hypomanic, or even mixed phase mania and depression than we can in healthy controls. Simply as we go from targeting the left VLPFC from targeting the control region. So again, evidence that we can actually change activity in this circuitry in people with bipolar disorder by targeting this left ventrolateral prefrontal cortical region. We've also looked at the EEG data during performance of the impulsivity task, the delayed discounting task that I was talking about earlier in people with bipolar disorder, and again showing a similar idea. Again, this is preliminary data also used as part of a proposal for which we're now very thankful to be getting funded by a nice foundation. And essentially, this is work with Fabio Ferrarelli, also in the Department of Psychiatry in Pittsburgh. And what we're showing here is that in people with bipolar disorder, before they have the stimulation to the left ventrolateral prefrontal cortex, they're showing higher beta power over the left ventrolateral prefrontal cortex relative to healthy controls. After the stimulation, we're showing that that power is vastly reduced, again, more so than the healthy controls. Again, indicating that we can, by targeting the left ventrolateral prefrontal cortex, damp down activity in this circuit in people with bipolar disorder relative to healthy controls, which is exactly where we want to be going. But then we're going to be looking at how this relates to mood, both acutely and longer term. And that will be the first stage to developing, and that indeed is the first stage to developing new brain stimulation treatments for bipolar disorder. So in summary, I just want to sort of highlight the sort of main take-home messages of this presentation, which I've been able to present to you today. And that is, we're using multimodal imaging to talk to you about fMRI, diffusion imaging, and also EEG to identify regions in the reward circuitry that predispose to bipolar disorder and mania, hypomania risk in particular. And this is, in particular, we're showing left ventrolateral prefrontal cortex during reward expectancy, as measured by fMRI here and EEG beta power here. This predisposes to bipolar disorder. We're also now using this as a promising neural target for new brain stimulation interventions to try and reduce the risk of developing mania-like behaviors in people at risk for and those already with bipolar disorder. And this, we feel, is an essential way forward to, even if we can reduce the number of medications that people take from 5 to 3 to 2, even if we can stop people needing, a lot of medications all the time, that would be a great step forward and reduce the side effects of these medications that many people have to take. And at that point, I am so grateful to you to listening to me today, and very grateful to my lab here. Some of them shown here. This was pre-pandemic at a celebratory party at my house, and I just want to thank all the people in my lab, and even those who unfortunately couldn't make the party at the time. I also want to thank the National Institute of Mental Health for funding me very nicely throughout the years with my lovely colleagues, and the Pittsburgh Foundation and the BBRF for funding me too. And thank you once again for your attention. Thank you.
Mary, thank you. And what a nice picture seeing your members of your lab together celebrate. Hopefully
Yes.
we're moving back toward that type of celebration as we move back toward normal.
I hope so.
Yes. So thank you for an excellent presentation and all the work that you and your colleagues are doing. And I guess two of the take-home messages that you're presenting relate to using these types of biomarkers for early diagnosis, and also using these biomarkers as potential targets for treatment.
Yes.
And we received a couple of questions. I just want you to clarify for people about some of these neuromodulation type of treatments. People ask, are these like mini shock treatments? And I want you to just respond to that question.
Sure. Well, first of all, thank you very much for the questions. So these treatments are not like the shock you're thinking of, ECT, obviously. They're not like that in that regard. They're much more focal, and they don't produce seizures as a means of treating. They basically produce a very discrete focal change in the electrical fields inside the brain in a very measured way, which is safe. And in doing so, change the way the brain is functioning abnormally. Now, I have to say that both of these treatments, TDCS and theta burst stimulation, TBS, TMS, as it's also known, TMS in particular has been approved by the FDA for the treatment of depression, so it's safe. And TBS is also approved. So ECT, of course, is a very valuable treatment for many people, but of course it's an end stage treatment because it does produce seizures, or can do. So basically, TBS and TMS are not about shocking people. It's really about changing discreetly the current magnetic fields and electric fields in the brain and changing behavior as a result of that. Often we will need to give these interventions for more than once. It would be over a course of a few weeks, 2 or 3 times a week, something like that. And that would be the plan. And then the idea would be that you set in train sort of more permanent changes in behavior.
Good, excellent explanation. And just to reiterate, really these treatments are not considered invasive types of treatment.
Correct. They're not at all invasive. I mean, basically you're putting a probe, electrode or a pad over someone's head, and there's no invasion into the brain at all. It just basically sits on the skull and the hair in the skull and goes through the hair in the skull. But as I say, in a very measured, discrete way, and resetting the balance, basically. That's the idea.
And the research that you're doing will help determine how to best use those types of tools by knowing where to direct the current to, as you described, to reset the brain.
Yes, exactly. I like to say that, there are treatments out there not for bipolar disorder, but for unipolar depression, which do, have a standard space in which to put the electrode or the pad. The problem is that one size does not fit all. We all are different. We all come in different shapes and sizes, and our brains do too. And so it's really critical to identify the right place in the brain for that person where you're going to put the pad or the electrode. And that's what we do in our studies. So we're kind of personalizing the targeting of these regions. So we're hitting the right spot in the right person at the right time.
Right. I think that's a very good point. Even now with medicine, there isn't a brain scan or a blood test in advance to know the right choice. You're working to really make use of biomarkers to help direct this type of treatment.
Yes, exactly. Yeah. Yes.
Yeah. And this is really a great example of how basic type of research can lead to clinical interventions that can actually help people. I want to ask you as a final question, where do you see this going? What's your hope and expectation for in 5 years from now, 10 years from now, where this work will lead to?
Well, thank you for that question, Jeffrey. I would very much like to see this work moving forward so that we can start to do full-scale clinical trials using brain imaging guided targeting. I mean, these trials are badly needed. People with bipolar disorder, there isn't enough research in this kind of work in bipolar disorder, sadly. It's because bipolar disorder is very challenging to study. It lags behind research in major depressive disorder and to some extent schizophrenia as well for that reason. So and that's where I like to see this going. But I'd also like to see more work where we dive deeper and really get down to the sort of finer tuned level of understanding of neural activity abnormalities in people with bipolar disorder. So we're using our imaging techniques, but partnering with basic researchers in other areas who are looking at non-human models, so we can actually really understand where the sort of neural level activity is abnormalities are coming from, and that will eventually allow us to develop even more fine-tuned interventions, non-invasive interventions, where we can target deeper structures with more precision than we can do right now. But I think the first stage will be to do clinical trials using that, hopefully building on the findings we're hopefully going to have soon from these 2 recently funded studies. So, but that's where I hope to see the field go.
Wonderful. Well, we're looking forward to seeing those results and the tremendous impact that it will have on so many people's lives. So, Mary, I want to thank you for all that you have done and continue to do and will do in the future. I'm so deeply appreciative.
Well, thank you so much, Jeff, and thank you for everyone who took the time out of their busy schedules to listen today. It's my honor to be here presenting, and honor to work with all the wonderful people and to work with the families and people sufferers of bipolar disorder, because that's the most critical piece. So thank you to everyone.
Absolutely. I agree with you. And I also want to thank everybody who joined us today. 100% of all donor contributions for research are invested in our research grants for scientists. All of the research we fund is made possible through supporters like you. So please consider making a gift by visiting bbrfoundation.org or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with family and friends, please visit the events and webinar page at our website. I hope you'll join us again next month when Dr. Gwyneth Zai, Assistant Professor in the Department of Psychiatry at the University of Toronto, will present OCD using genome data to predict risk, symptoms, and treatment response. This webinar will take place on Tuesday, December 14th at 2 p.m. Eastern Time. Once again, thank you for joining us. We appreciate your ongoing support. Take care.